This video will be part one of changes in chromosome structure. Chromosomes can be altered in several ways. We bend these alterations into the four broad categories you see here: deletion, duplication, inversion, and translocation. Each of these has the potential to have phenotypic consequences, but some are more likely than others to affect the phenotype of an organism. Shown here are examples of specific changes in chromosome structure. The first one we will talk about is a deletion. This is straightforward to understand, as a deletion is simply a loss of genetic material on the chromosome. If this happens on the end of a chromosome, it is called a terminal deletion, and in the middle of a chromosome, it is an interstitial deletion. Deletions often have phenotypic consequences, and these are typically negative in nature. One example is Cri-du-Chat syndrome, deletion in the short arm of chromosome 5, which leads to a phenotype of a cat-like cry in infancy, short stature, characteristic facial abnormalities, and microcephaly. Duplications are when a region of DNA is duplicated in a chromosome. Whether this duplication has a phenotypic consequence is largely based on the size of the region that is duplicated. The larger the duplicated region, the more likely it will cause a change in phenotype. Duplications typically arise during abnormal recombination. This can be from the misalignment of chromosomes during meiosis, followed by crossing over. This is often due to repetitive sequences. Phenotypic consequences of duplications are not always bad, though. Duplication events explain the origins of gene families. A gene family is two or more genes that are similar to each other. Once duplicated, the selection pressure on the extra copy of the gene is reduced, and this can allow it to evolve to have a slightly different function. A great example of a gene family is the globin gene family. Over 500 million years, this gene has been duplicated many times, and there are now 14 paralogs of this gene in our genomes. Each version of the globin gene has a slightly different role in binding oxygen. For example, myoglobin stores oxygen in muscle cells, whereas hemoglobin binds and transports oxygen via red blood cells. Duplications result in copy number variation among our chromosomes. So with a genome, a particular gene may occur more or fewer times on a particular chromosome due to duplication events. This is surprisingly common, and in humans, the rate of copy number variation among our chromosomes is about 0.4%.